All right, hello everybody. We're going to be moving on to topic 6.2 today, which is going to be on transcription and RNA processing. So before we start talking about this, a term you're going to hear a lot is this idea of the central dogma of molecular biology. A lot of times this likes to come up in questions, and essentially what this means is one of the most fundamental rules or informational pieces we have about molecular biology is the idea of how proteins are assembled. And this is essentially going to happen over two different processes. You have transcription, where RNA is going to copy the gene that we need to make the protein from. And then you have translation, in which the RNA strands are going to be translated from RNA into the actual proteins themselves through the use of different molecules. So whenever you hear the central dogma, essentially what that refers to is this idea of how we go from information in DNA to a finished protein or polypeptide. Now, in order to do transcription, which is the process that we're going to be talking about today, and translation, which we'll talk about this weekend, there are three major RNA pieces that we're going to need. The first one is the messenger RNA. This is a strand of RNA that carries DNA information to the ribosomes. Your cell doesn't want to take the DNA out of the nucleus to the ribosomes where the proteins are actually made. So we're going to have kind of an in-between person here called messenger RNA, which you can see right here is just a single strand. Then we're going to have transfer RNA. Transfer RNA is highly folded molecules, and essentially it's made up of two main components. At the top here, we have an amino acid, which is going to be used to construct the protein we're trying to make. And then on the bottom, we have an anticodon, and this is what's going to translate from the messenger RNA into an amino acid. So you can kind of think of it as an adapter that you kind of go from codons or genetic information into actual amino acids. And then the last one is going to be ribosomal RNA, also known as rRNA. And this is essentially going to be folded and manipulated with proteins in order to build the ribosomes themselves. And hopefully you remember from Unit 2, ribosomes are the organelle that is responsible for protein synthesis. But also, this RNA is going to be used as a catalyst, also known as a ribozyme, which is going to help speed up translation so we can make proteins even quicker. Now, in prokaryotic and eukaryotic cells, proteins are necessary, so transcription is going to happen in both of them. In eukaryotic cells, transcription is going to occur in the nucleus, while in prokaryotic cells, it's going to occur in the general cytoplasm area. Now, the first step of transcription is you're going to bind RNA polymerase to the non-coding DNA region called a promoter. So in order for RNA polymerase to start making the mRNA, it needs to know where to start. And so what we have is a non-coding region, also known as like non-important information. You can kind of think of it like if you use sticky notes in your notebook to mark certain parts, or a bookmark to mark certain pages if you're reading a book in ELA. That's kind of what this promoter region is. It's a region for RNA polymerase to recognize and bind to, and then actually start transcribing the important information that goes into the actual protein itself. So you can see the RNA polymerase here splitting that DNA molecule and beginning to read it. Now, transcription, as I said before, does begin with the binding of RNA polymerase to our non-coding DNA region. Now, typically, this promoter region is going to be called a Tata box in eukaryotic organisms. We call it a Tata box because it's a region in which T and A bases are repeated over and over again. So it's a very common example of where this RNA polymerase begins. Now, RNA polymerase isn't going to work alone in order to identify and help speed up transcription. There are going to be other proteins known as transcription factors. So transcription factors meaning they help with transcription. And what they're going to do is they're kind of assist RNA polymerase in finding that promoter region or those Tata boxes in order to know where to start. We'll talk a lot more about transcription factors later on in the unit, but just kind of keep that in mind that transcription factors are going to play a big role in telling the cell which proteins need to be created and which ones do not, because every cell is going to have a unique list of proteins that it needs to make to carry out its unique functions. Now, RNA is synthesized from the template strand. So you can see here RNA polymerase has split the DNA into two. The one that we are essentially base pairing with is called the template strand, and it's going to be synthesized from the 5' to 3' direction, which is the same as DNA replication. Now, template strand is what I'm going to try to call it this entire unit to keep it consistent. But just so you know, if you're reading questions, you might see words like the minus strand, the non-coding strand, or the antisense strand. So there's a lot of words to kind of dictate the name of the strand that the mRNA is base pairing to. But I'm going to try my best to say template strand for all future videos and class activities. Now, once the RNA has completely read the region or the gene that we're trying to make, or make a base pairing of the template strand, we're going to have something called pre-mRNA. We call it pre-mRNA because it's not quite ready to be used to make the protein. There's going to have to be certain modifications that need to be happening in order for it to become mature mRNA. The first one is we're going to have to remove certain segments of the RNA called introns. Introns are represented by the white regions here. So some of the regions we don't need to make the protein, so we're going to cut those out, and we'll talk about that more later. Then we're going to add something called the G-methyl cap, which we see right here. This is going to help assist with getting the mRNA out of the nucleus and keeping it protected as it works its way to the ribosome. And then at the end, or the 3' side, we're going to have a poly A tail. And the poly A tail essentially is a protective measure on this side. That way, if we lose base pairs, we're not losing anything important. We're just losing some of this poly A tail overall. So here's that splicing that I was just talking about. And splicing is going to be carried out by something called spliceosomes. And spliceosomes are just small bits of RNA that help remove the introns. So again, these introns are regions of the mRNA we don't need for the protons. Exons are going to be regions that we do need. So these spliceosomes are going to come in, cut out the introns, and then splice together the exons. Now, once we have our G-cap, our poly A tail, and our introns removed, then we have a piece of mature RNA that is ready for translation, which is the next step. Now, which part is going to be cut out and which part are going to be left in is going to be unique to the protein and the information that it needs. Now, what's kind of cool about mRNA is that we don't always have to splice it in the same exact way. We can splice it in unique ways in order to create different proteins from the same information. So, for example, you can see in the top here, we have a DNA region, and we transcribe the entire top part here. The parts that are not glowing, these are introns, and they're going to be removed no matter what. The parts that are glowing are different exons, and they're numbered 1, 2, 3, 4, 5. And we can use different regions and splice them together differently in order to make unique proteins. So for example, if I wanted to make protein A, I'm going to splice together region 1, 2, 3, 4, and 5. If I wanted to make protein B, I'm just going to splice together 1, 2, 4, and 5. I'm going to leave 3 out. I'm going to cut 3 out. And since I left this whole segment of information out, I've now created a totally unique protein from the same RNA I've had before. And you can also do it for any other number. So in this case, we cut out 4, and we put together region 1, 2, 3, and 5. We were able to get another unique one. So from the same mRNA piece, we can create multiple proteins depending on which exons we want to keep. So please remember, we always get rid of introns, but we do have selective, choosing which exons we want to keep for the protein. All right, and the last slide here is going to be another one of these animation videos. I do recommend that you watch it. I'm not going to go over all of it here because it pretty much just goes exactly what I just went over, but I will go over it tomorrow in class. But if you would like to watch this video, just pull up the notes on on Google Classroom, and then you can watch it and kind of see if you can apply everything we talked about here.